Namaste students. In our previous module, we saw something about how do organisms reproduce, and we went on a safari into the jungle. We saw a lot of organisms with their young ones. Of course, we also concluded something about the asexual reproduction and sexual reproduction. We were just getting introduced to that topic that there will be some variation seen. Where do you observe more variations? In asexual reproduction or sexual reproduction? You're absolutely fine. A lot of variations will be observed in the sexual reproduction, whereas in asexual reproduction, we do not observe that much of variation. Why? Asexual reproduction involves only one parent. So there is not going to be any formation of sex organs, or there is not going to be any fusion of gametes. The young ones formed are almost identical to each other. The reason is they receive all the genes from a single parent. This asexual reproduction, we observe it in unicellular organisms and in multicellular organisms. The unicellular organisms such as bacteria and protozoa, the multicellular organisms like algae, fungi, planaria, hydra, they also show the asexual reproduction. When we see this, we find the asexual reproduction can occur in two conditions. It can be under a favorable condition. It can be under an unfavorable condition also. Depending on the situation, the organisms try to change themselves, adapt themselves, and start reproducing. We quickly check the various modes of asexual reproduction. The various modes that we see in the asexual reproduction is going to be fission, fragmentation, regeneration, budding, spore formation, vegetative propagation, and propagation by the tissue culture, of which in this module we are just going to focus on the 5 important topics of fission, fragmentation, regeneration, budding, and spore formation. Coming over to fission, when we just use the word fission, what do you understand by the word fission? Correct? It's just breaking down. So we find this is one of the common asexual reproduction that we observe under the unicellular organism. It involves splitting of the parent cell into 2 or more than 2 separate daughter cells. Now based on 2 or more, we try to classify them as binary fission and multiple fission. If we say binary, by the term binary, we can understand one organism is just getting split into 2. By the term multiple, we can understand one organism is going to get divided into many. Let's try to focus a little more on that and try to understand. Here is a small video about binary fission in amoeba, where we find this amoeba being a unicellular organism. It is a type of asexual reproduction in which a fully grown parent cell splits into 2 halves, producing 2 daughter cells. What happens first? The very first step is they have a lot of pseudopodia because they are irregular in shape. They combine and try to form a spherical shape. Slowly you observe the nucleus dividing. We call that as karyokinesis. And then you observe the cell membrane also slightly showing a constriction and trying to divide the 2 cells over there. And this division of cytoplasm is what we call it as cytokinesis. 2 daughter cells are formed. They also look exactly like that of the parent cell. Not much of variations over here. So that is what we observe in the case of binary fission in amoeba. Let's just quickly go over to the notes of binary fission. What exactly happens? I repeat it, we recollect. Amoeba reproduces by binary fission only in highly favorable condition. It needs all the conditions to be favorable. Moisture has to be there, the temperature has to be perfect, then only you find that amoeba will be able to divide. So amoeba, first I told you, the pseudopodia is withdrawn. They are irregular in shape, so all the pseudopodia is withdrawn and forms a spherical shape. Then the nucleus of the parent cell divides, we call it as karyokinesis. The cytoplasm divides, we call it as cytokinesis by the constriction of the cell membrane. Then what happens? the mother cell divides into two cells, each containing a nucleus, resulting in the formation of two daughter cells, which are going to be 100% replica of the parent. The reason behind it, they're not going to show that much of a variation. The variation we are going to observe in the case of asexual reproduction or in the case of unicellular organism, it's very, very negligible. Fine. This cell division occurs in any plane in the case of an amoeba. The reason behind this, amoeba is having an irregular shape. It can just divide at any plane. There is not needed, it is going to divide only at a particular plane. But what happens? Let's go and check about another organism which also undergoes binary fission. Binary fission in Leishmania. This is an organism which causes Kala Azar, that is, gives a temperature, fever, and also sometimes swelling in the liver, in the spleen. That sort of a disease is caused by this particular Leishmania. And they have a whip-like structure, in the sense they have a very definite shape. And because of the definite shape, you find the binary fission occurs in a very definite orientation. It has to just divide into two. The reason is the relation of the flagellum over there, and it has a definite shape. That is what we see in the case of a binary fission. We'll now move over to multiple fission in Plasmodium. This mainly occurs when it is going to be an unfavorable condition. What is Plasmodium? Plasmodium is a malarial parasite. Most of you know about it. What exactly happens? This Plasmodium, during the unfavorable condition, the parent cell starts a thick resistant wall around it. It's like a cyst. Protects itself completely. And then you find within the wall, the nucleus starts dividing many times and try to arrange themselves in the peripheral region. That is during the maturity stage. Once they reach over to the peripheral stage, you find that the cytoplasm slowly starts dividing and keeps surrounding each nucleus. Whatever the number of nucleus are going to be divided there, that many cells will be formed. And when the condition becomes favorable, the cyst wall will break. When it breaks, the Plasmodium are released. There will be many number of Plasmodium. Because it is many number of Plasmodium, that is why we call it as a multiple fission in Plasmodium. We have been seeing about reproduction, asexual reproduction, especially in a unicellular organism. Let's quickly start observing the next level. Here we are going to see something about Spirogera. It is a multicellular organism, but it is not reached to the tissue level. They are cells just arranged by the side. And they are algae found in the water. What exactly happens? When they are present in the water, because of the water current or something, you find they can break down into smaller pieces. Or in other words, we call each of the piece to be as fragments. And what exactly happens? Each fragment has the capacity to form a new individual. When they're just getting cut here and here, you find lengthwise they start growing on top as well as at the base. They are continuous ribbon-like structure. As a result, fragmentation takes place over here. And therefore, we find it's more or less, when we talk about fragmentation, we move on to the next topic, which is regeneration. That is also considered when one organism is divided into many pieces. Each organism can grow into an individual. Then the question arises in our mind, why can't it be a fragmentation? Why can't it be related to that of a fragmentation? When we talk about fragmentation in Spirogera, you find they have not reached the tissue level or an organ level. But in the case of a planaria, you find that the cells are divided, they have become to the tissue level, and then you find then they also have come to the organ level as such. That is what we find in planaria. Regenerations are supposed to be a specialized cells which are found all over the body in planaria. So when they are just going to be divided, you can see the diagram over here. When they're just going to be divided, whatever the number of pieces, you find them getting divided, each part will start growing the other part. That is what we see in planaria. So they grow and develop. What exactly happens? This particular situation, when they just start getting divided, the cells which are present over there, they start multiplying more. They start proliferating, means arranging themselves and developing themselves into the various tissues and organs. As a result, you find a complete individual will be observed in the case of a planaria. We'll just have a small look at a video which shows the regeneration in planaria. We just observe planaria. If the planaria has been cut into two equal halves in this manner, we find that the bottom as well as the top is being developed into two. In case if the planaria is cut vertically, you find the other half also grows. So that is what we observe in planaria. In case if it is cut half, we also observe the upper part of it alone is being divided, whereas the lower part is not getting developed into another individual. So these are the various types of regeneration that we can see. It can be either cut into equal halves horizontally, or it can be cut in a vertical position, and still you find that the planaria keeps growing. But sometimes we find this regeneration cannot be considered into a reproduction process, especially when you're going to talk about a lizard. You find in case the tail goes off, it is just going to be only the growth of the tail and not a complete individual over there. We move over to the next one, is budding in hydra. In hydra, we find regeneration is possible. Why? Because you find the regenerative cells are all over in the case of hydra. But hydra shows another unique division also, which we call it as budding in hydra. What exactly happens? A very small outgrowth appears on the parent hydra, and this outgrowth starts repeating the cell division so many times at one specific site. As a result, they start forming a bud. Slowly you observe the bud growing into a young individual. Once it grows into a young individual, it can detach from the parent and it can start growing as an individual organism. It will be very interesting if you're just going to quickly watch over to the video how budding in hydra takes place. Here we see hydra, which is an aquatic organism, and it is just present over there. You have the tentacles for them, and they have an opening, oral opening, and slowly you can observe the bud developing. Can you see the bud is just developing into a young individual? And as it gets developed, they can try to detach themselves from the parent, and they start growing into a young hydra, which looks exactly like that of the parent, and that is what we call it as the asexual reproduction, that is budding in hydra. Budding is not only seen in the case of multicellular organism like in the case of hydra. It can also be observed in the unicellular organism like yeast. Most of you will be knowing more about yeast because in your house, your mother, if she is making some food, like if it is going to make appam, or in the bakeries you find they're using it for the bread making, definitely they'll be using yeast, which is considered to be a unicellular organism. So they can reproduce much faster. What exactly happens? You find in the case of the yeast, the parent cell, a small bud-like structure outgrowth comes over there, appears at one end. Slowly you find the nucleus divides and starts moving into that. Then immediately the cytoplasm divides there, and you find the bud with the nucleus is present over there. The parent cell, yeast is here, and the daughter yeast is also there. Slowly this daughter cells will start maturing, and that will start dividing, and they try to form a chain of yeast. This is how they multiply much faster in the case of budding in yeast. We have been studying about yeast is helping in the formation of bread, and now we're going to study something about spore formation, which is going to spoil the bread. I'm sure most of the time in your house you try to buy bread, we don't consume the full lot and we try to leave it aside. A day or two or three, after that slowly you'll start observing some black spots. What exactly happens? Spores are something which is the spore of the rhizopus, which is found in the atmosphere. And when the substratum becomes comfortable, suitable for them, they fall onto the bread, and slowly they will start forming a very mild hyphae, a thin hair-like structure, white color. Sometimes we don't observe it also. We do not observe and we try to consume, and we do get sometimes food poisoning. So this hyphae slowly starts developing more, and they start penetrating into the bread. And some of the hyphae will start giving up vertical stem-like structure, and we call that to be as the sporangiophore. The end of the sporangiophore is what we call it as sporangium. Sporangium is where you find the spores will be present over there. These spores are going to be innumerable. You find they just keep increasing. And what is the stage that we are going to see? When it almost becomes black, that is the right matured state. You find that the sporangium just burst. Immediately the spore comes up, some gets carried away in the air and some falls onto the bread again. That is why you find that the fungus formation over there increases. It is just not in bread, because in bread we call this to be as a bread mold. We do observe this in many of the fruits, and we also observe sometimes in the leather goods and other places also the fungus infection can keep growing over there. So this spore formation, the spore which is being formed is going to be tough, really tough, and it is going to be a resistant structure which are dispersed by breaking of the sporangium. So when the climatic condition becomes favorable, you find they multiply more. What exactly happens? You find that they need 3 important characteristic features or situations for them to multiply more. The first one is going to be a suitable substratum, like in the case of a bread. Bread forms a suitable substratum. Second, there should be an optimum temperature. It should not be extremely cold, it should not be extremely high. A right amount of temperature is needed. Third, it definitely needs a moist surface. You can try out this experiment. If you just keep a dry bread aside, the formation of bread mold may be there, but it will be a slow reaction. But if it is going to be a moist surface, you find that the formation of the spore is going to be much faster. We'll just quickly have an observation of the next video about how we can make the spore formation. Let's observe the bread mold fungus in the lab. We are just going to take 2 slices of bread. We pick up the first slice, slowly dip it in the water, that gives you the moisture, and keep it on one plate. Then we take the next slice and we keep it on the other. There will be some moisture content in that. From the air, the spores are actually falling onto it, and you find that the fungus slowly starts developing. And that is what exactly happens over there. What exactly happens? You find that there will be hyphae which is moving inside, and these hyphae which is moving inside will go deep into the bread, and they will start giving nourishment for the sporangiophore. And in the sporangium, the spores will be there, and this spore, when it matures, it breaks and then it gets dispersed in the air. That is how you find that the spore formation exactly takes place. We take up a question: why multicellular organism cannot go for a cell by cell division? When we talk about multicellular organism, by the term multicellular organism, we can understand the cells are going to form tissues, tissues are going to form organs, organs are going to form organ system, and they are all well placed inside the body in definite positions. So when it is going to be in a definite position, what exactly happens? If it is going to be a cell by cell division, like in the case of amoeba, or it is going to be in the case of a leishmania, it's not going to be possible. All the organ systems are not going to be evenly divided. That is the only reason we find it is not going to be possible in the case of a multicellular organism to undergo the cell by cell division. Fine, we just studied something about the asexual reproduction. We just quickly move over to a quiz about what we have studied, what we have understood in this particular module. Which among the following is a unicellular organism that reproduces by budding? We have Hydra, Planaria, yeast, and Spirogyra. But it says a unicellular organism reproducing by budding. Right, the answer is the yeast. We go to the next question. The process of division of cell into several cells during the reproduction in Plasmodium. We saw something called fission, cell division, right? So in Plasmodium, what sort of a fission has been observed? Is it fragmentation, budding, multiple fission, or binary fission? Right? The answer is multiple fission. We'll go to one more question. The rapid spreading of bread mold on the slide of bread are due to, I've given 4 options, and then I've given A, B, C, D, which has 2 options each. So we have to just read through, try to understand, and pick up the right one. It says the presence of large number of pores in air, Correct. The presence of large number of thread-like branched hyphae. Yes. Presence of moisture and nutrients, formation of round-shaped sporangia. We move over to the second question. The process of division of cell into several cells during reproduction in plasmodium is termed as, let's quickly recollect, fragmentation, budding, multiple fission, binary fission. It clearly says reproduction in plasmodium with several cells. So the answer, perfect, it's going to be multiple fission. So let us stop this module over here in asexual reproduction. We'll continue asexual reproduction in the next module. We'll be studying about the vegetative propagation and tissue culture. Namaste.